So, welcome everyone. I'm Alexander Mihalcea, and today we have of one of the industry leader, Marcin, CEO and co-founder of the Real Research. Hello, Marcin. How are you today?
Hello, Alexander. Nice to meet you. Thank you for the privilege. I'm actually really good, and I'm excited to join this podcast.
Yeah, awesome. And I'm excited to have you too on the podcast today. And I'm sure you're going to be of the great value for the audience.
Thank you very much.
Now, before we dive in, could you give us a quick overview of what Real Research does?
Yes, sure. So at Real Research, we are doing hydrogels for 3D cell culture. That is really the product of the company, but you can look at it from a slightly different perspective. So basically, um, our product is a tool that helps companies that develop new drugs do it better, faster, and much, much more efficiently. So we are basically one of the components, one of the value chain of drug development.
Yeah. Okay, that's really fascinating. And what inspired you to create Real Research? Was there a specific moment or experience that
Yeah.
sparked this journey?
Yes, sorry, I had something in my eye. Yes, it was actually really exciting and interesting to us, me and my co-founder, because I was doing my PhD and she was doing her master thesis, and at that time, we had another collaboration which kind of worked. There was some interesting things. I mean, scientifically interesting, but we were responsible for making the gel, and it was a kind of very friendship related collaboration. And afterwards, we realized that this is actually something much, much more important. And
Yeah,
we thought just about publishing, but that wouldn't give us the patent. That would be very, very hard to do at the university. And then we really thought that we have to see whether that's just another publication or we can build something on it. So there is in our industry there is something called angiogenesis assay. It's basically an experiment that can tell you whether the cells can produce blood vessels. And so far, it has been done only with one product, with one competitor. So we thought if we could meet this standard of being able to do the angiogenesis, say we should start the company.
okay.
And it was really interesting because it took us like 6 months really to get there. We invested our own money. We failed. Like we literally had the last 50 milliliters of all the ingredients necessary. And it was really the last experiment possible. If it wouldn't work, we just wouldn't start, and the history would be very different. And surprisingly, we were supposed to do this experiment during the weekend, but it was a May summer nice weekend, so we
Yeah.
didn't do the experiment through the weekend, and we did it on Monday, and it worked. And we didn't know why it works, but it really worked. And then we obviously we bought all of the new ingredients we needed for the
Yeah.
experiment, and again it didn't work. It took us another 3 months to get to learn what happened during the weekends and why it actually worked. So it took us another 3 months to repeat the result, and then another probably like 6 months to get to the right protocol, how to make the gel. And then we thought, well, that's enough, and we started the company.
Yeah, interesting things. And what was your background before starting this venture?
So this was actually very much aligned with what we do. I was doing my so I studied biotechnology as my master. Then I was doing my PhD in cancer immunology, and actually new therapies of cancer, like the biological therapies of cancer. And during that time, I got a scholarship from the Polish Foundation of Science. And during that scholarship, and within that, I got also the business training in Judge Business School in Cambridge on the program is called Ignite. I highly recommend it. And then when we already started the company, I wanted to refresh all of that, and I also joined the Maxwell Center Impulse program, also in Cambridge, which is really fantastic
Yeah.
people, and great experience to learn how to do business if you have the science background.
Okay, and I have a curiosity by then because you mentioned this. So, what were the biggest challenges you faced in transitioning from a scientific role to a leadership position?
Yeah, um, to be honest, um, I see two questions in this question because from, because from scientist, transitioning the challenge of transitioning from a scientist to business is you really like in science there is a lot of emphasis on the data, and although it's not really true, but there is this feeling like the data will defend itself. It's not really true, but this vibe exists in science. Whereas in business, my observation is the other way around. It's really the way you present different things. It's really it's much more about the connections you have. It's much more about those much more how to say that socially related arguments that influence
Yeah.
your success or failure. Like with a podcast, I'm also watching different podcasts, and when once you become more famous as a podcaster, it's much easier for you to get more other people,
Yeah.
not only as viewers but also as guests. That obviously increases your visibility, and I feel like that's how business works, and to some extent science as well. Because if you do a PhD in a very well-renowned laboratory, obviously that's going to help your career. But in science, there is yeah, the emphasis is really more on data. So eventually, if your data is really solid, in theory, you really can come out of
Yeah.
nowhere. Where in
Right.
business, it's much less so, I would say. But in terms of the challenges, um, more on the market of what we do, it's really more the challenge of the market where that we are really facing, and that was also the challenge movie. Like in science, you think of being more again, the data will defend itself. Whereas in business, it's very different. So the challenge that we are facing, like we're trying to solve as a company, is basically extremely high failure rate in oncology. So 97% of drugs that get to phase 1 clinical trials in oncology fail, and
Wow.
it costs like $1 billion to get those drugs to this phase 1, and then you have 97% failure
Yeah.
rate. So within this 97%, half of that is due to toxicology. So this drug is simply too toxic and would kill patients anyway. But the other half is efficacy. So the question whether the drug actually works or not. So
Yeah.
drugs can work perfectly in animals, or in vitro or in the lab, but then fail tremendously in clinical trials. And that's the other half of the equation. And what we are really trying to do, and I think we have good evidence that we will be able to not entirely, but significantly improve the statistics, is we can really work on the efficacy side. Because we see
Yeah.
that the research models we are creating have this extremely high translatability to clinical results. So that would
Right.
be
Right.
really the two challenges I saw in your question.
Yeah, yeah, yeah. It was very interesting one. And because you mentioned this, and this is a very interesting point, so let's go back to when you first started. So,
Mhm.
what major challenges or gaps did you see in the market that made you think this needs to be solved?
Yeah, so to be honest, it's so what we're doing is a three-dimensional cell culture. And if you look at, if you look in the mirror, or anybody watching this looks in the mirror, we are three-dimensional. Like
Yeah,
on our website, you can even see there is a one cartoon where we saw like when we kind of flattened a human, and we say
right.
that's not natural, and that's really true. So what we saw on the market is that like 95% of all cell culture is done in 2D cell cultures, which there is some use for that, but it's certainly not when you want to test whether the drugs work or not.
Right.
So I'm saying this because there is this 5% that is already in 3D cell culture, and we asked ourselves this big question: why it's only five, since it can be so much better. And really, the struggle is that they are very dispersed. So you can think about it like like in the early years of electricity, you didn't have simply the socket. You actually have the
Yeah.
socket. You had vacuum cleaners that you had to screw into the socket. There were no standardized solutions. Now everybody uses, like in Europe, it's basically the same. Okay, Asia is different, US is different, but they are standardized to that economy, to that part of the world. Whereas
Yeah.
in 3D cell cultures, it's all around. So you have suspension 3D cell culture that has some upsides but many downsides. There's one of our major competitors that has many downsides, but the upside is that everybody knows it. And there are a few other technologies that try to overcome it. But if you look from the very, very high level perspective, all of that is based on basically four different ideas. So one is suspension, so no scaffold, no ECM, no gel, basically. Another is a like protein-based
Hmm.
gel, which is also part of what we do, but I will give you a correction in a second. And then there are like non-protein gels, and then on top of that, you could also think about like very stiff scaffolds. And the first one is very easy to do, but has the lowest value. So
Yeah.
suspension, like it's no attachment. There are applications where this makes sense, but there are very few of them. But it's easy and it's fairly cheap. So that's another another argument. Then the
Right.
second one is actually the one that has the most potential, and where our bigger competition is. But the product they have is not repeatable. It's very expensive, very hard to scale, and is produced in mice. So they just have tens of thousands or even hundreds of thousands of animals to produce that. And then the second one is simply it's made of trees. So there again there are some applications, but it's not really working. And the
Yeah.
fourth one is actually only, in my opinion, it would be good only for like bone cancer studies. And I know I may be going to some boring details, but I'm
No,
saying
no, no.
this
I see.
because this business is very dispersed. That's really the challenge. And what I think we can do quite uniquely is we put all of that together, and you can do all of those things even better in our solution. And on top of that, it's actually like it's comparable. So you can do very first studies with our system. You can upgrade the system. You can then do the second study, and you can get from very early stage up to the clinical trials using the same comparable data, comparable like research tools. And
Yeah.
that's
Right.
that's really the uniqueness of our approach. That in terms of the product, and on top of the product, we've built quite a lot of validation of our research models. We show that our research models have this very, very high translational potential. So what we see in lab is almost like it's never identical, but it's
Hmm.
extremely close to the drug responses you can see in patients. So that's really the uniqueness and the challenge, that the bigger dispersion problem. And with that, we also hope that this 95% that is using 2D cell culture will obviously be part of will use our solution in long term.
Okay. And for example, I have a curiosity that you mentioned here. So, what are the key reasons for the low success rate in oncology drug discovery?
So to be honest, like from like one is obviously just the complexity in biology. So this is something nothing nobody can deny, and I will not try either. But there is one, and this is also part of the name of our company. So I get sorry. I will ask myself a question, and then that will lead to your question. So very initially, like our product is called LifeGel, and very initially we thought about how to call the company, and my co-founder came up with the name Real Research, and
Yeah.
it came from a very very famous study. There is this there's one of the big pharmas has done a study where they wanted to just repeat. There was no research, just repeat studies published by in top journals, and they. By the way, can you guess how many results they were able to repeat? They took 67 publications. So how many would you say they were able to repeat?
2%?
Okay, you're good. It's five.
Okay.
But it's still extremely low. It's
Yeah.
like
Yeah.
science is about repeatability. If there is something that works, you want it to work. When you buy a drug, it should work. It
Yeah.
shouldn't
Right.
be 1 in 20 or 1 in 100. It should work. So 5% is extremely low. By the way, in the first experiment, they used only the protocols that were in the publication, and then they called the authors of those publications, and they were able to increase that, I think, from 5 to 15, something like this. But basically, it was still significantly below, below 50%, and that, it's. I think an acceptable number would be like you could repeat 95%. I mean, the reverse would be maybe acceptable, but
Yeah.
it was so striking. So our goal was really to make this real research. So that's where the name comes from. So going to your question, I think one of the major problems is exactly that. So uh, people are using very different research tools without standardizing them, without a good validation, and very often when you this is also one of the reasons why we think we have a huge upside. We are bringing huge upside to this market. Is that very often this drug development process starts with very large library of compounds of
Yeah.
potential drugs. So you have, let's say, you have 10,000 or 100,000 different substances that you want to test against a potential new target that you can have in the cancer cells. And this is called high throughput screening. So you can do this high throughput screening on 2D cell cultures. And this is really interesting, but it's also that's also a big part of the problem, is that when you do this in 2D cell cultures, you will find most of the results will be false positives or false negatives, mostly false positives, meaning the drugs that will look like they work will not actually work in clinical trials. So you
Yeah.
use the wrong system to pick the wrong drugs and then invest hundreds of millions to eventually fail in phase one. That's really the biggest issue in this market. There are some false negatives, which are very interesting. There are some examples on the market, and we actually were able to pick up one as well with our system. But really, the biggest challenge is are the false positives, because you can easily choose the wrong drug. That's why
Yeah.
that's what I meant with the validation. So what we're doing, just as an example, I will give you an example of one very commonly used drugs. It's gemcitabine. It's used against pancreatic cancer. So when you used 2D cell culture models, the concentration, the working concentration, so something you should think about giving to the patient, is 700 times lower than the concentration that actually works in patients.
Okay.
And when we did this in our 3D models, we are only zero point 0.5 lower.
Oh.
You see, we're almost in that exact concentration that you should give to the patients. So you could you could basically, there is a part of drug discovery process called uh, dose escalation, where you have one dose that you think could work or not.
Yeah.
Like that's a still question whether it's going to work. But then you want to see what is the tolerable, the dose that the patients will tolerate. Obviously, you don't want to go too high, but you want to know how high you can go. When then you will go to the efficacy studies, mostly in phase two. You know what you can test and what you should not.
Right.
So I'm just using this as an example, because since what we have is just 0 point, it's almost to the spot.
Yeah.
It's so close. So I'm saying that because there is this huge problem that you can easily pick up the wrong drug and run with this for 5 years and fail in phase one. So what we think is much, much better to use much tougher tests very early, when you still have, a few hundred compounds or even a few thousand compounds. And once you do that, it's important to keep in mind that very few drugs actually pass the assay, the test that we're performing. But those that do pass the test have, the biggest
Yeah.
potential.
Yeah. Yeah. Yeah.
That's really
Right.
the
Right. Right.
value, and that's really the reason. Obviously, there are a few other reasons. People from the industry would probably add something to this discussion. I'm just saying, like, more from my point of view, as somebody who is really focused on making in vitro models. Obviously, in vivo is slightly different, and then clinical trials. People from clinical trials would basically have probably some other also valuable arguments, but to another level
Yeah.
of drug
Yeah.
development.
Right. 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 I see. It's such a big complexity here.
Exactly, exactly.
Yeah, yeah. And for example, when you talk to potential clients or partners, what's the
Yeah.
biggest misconception they typically have about what you do?
Oh, there are a few. I actually just spoke with one who like he totally understood what we do, and he totally saw the value. But it's also another, it's just again another challenge. It's like for him it was really interesting. It was really interesting for me that he is also looking from the perspective of the regulatory process. So the regulatory process is actually still looking, is still accepting these very low quality two-dimensional cell culture assays. And that's really one of the major challenges, because um, for those companies who even would like to do something in 3D, they still have to do also some they would like to do in 3D. They still have to have some data in 2D cell cultures,
Right.
because that's what the regulatory system actually accepts. So it's that's really, but it's also like, I think it's also a very very good moment for us to be in this market, because it was really like for the last year or two, FDA, so the Food and Drug Administration in the US, was pushing very, very strongly to more advanced
Yeah, yeah.
vitro models, and that is in our industry, that's really huge, because they basically set the bar on a level that it's like, we obviously think that's the correct, we as a people in the industry think that's the
Right.
correct direction. And I'm not sure if I'm not mistaken exactly, but I think something like a month ago, they actually said that they will not grant any grant funding to drug development that will be done only with animal models. So you have to include some advanced like mostly 3D in vitro models, and that's
Yeah,
really
right.
good, because you have to keep, like, you don't have to, but I think it's still wise to keep some animal models, because they have some other applications, although still questionable. But like, I think the industry is not advanced enough to take over everything, but there is a lot already. And I think that, especially from our perspective, especially in the efficacy studies, there is this huge misconception that it used to be for a very long time a nice to
Yeah.
have, not a must have. But I think the FDA thing could actually make it into a must have. So that's a very, very good thing for us. Um, and um, and there is another misconception which we are also trying to fight, and I mentioned that somewhat in a few minutes ago. So 2D cell culture is easy. It's like you can teach a student in an hour how to do it. Obviously, to do it correctly, you have to have 20 years of experience. But to do it it's really, it's maybe I exaggerated that, but it's really quick and easy. I think I could teach you, if you know how to type it. I could teach you that in a few hours. But to do correct, well designed 3D cell culture is actually much more complicated. But on the market, you can see there are some companies that are doing those extremely complicated models. Where to be honest, I think they are too complicated, because they are very low throughput. They are scientifically very interesting, but they are extremely low throughput, extremely expensive. Like like testing one compound can easily cost you several $1 million. And the misconception is that there are those two extremes, or you can go very cheap, very easy, but with huge failure rate and a lot of false positives, or you can go to those very extreme, extremely expensive, but more valuable. So my understanding is the best is in the middle, and I think that that's where we position ourselves. So we're obviously much more advanced than the 2D cell cultures, and this is the data I mentioned about gemcitabine, the concentrations and how it works. It's really like it works that was done on pancreatic cancer. So that's what I can really speak with confidence about. But those extremely, extremely complex models, on one hand, they do deliver loads of very interesting information, but it's more art than science. Because to do this, I know one company, I unfortunately I can't give you the name, but to do it, they have a lab with like 30 technicians, where they want to source every single cell type primarily from the same patient. And I'm not talking about two types of cells, I'm talking like 8, 9, 10 types of
Yeah.
cells. And all of them have to be extremely fresh. And that happens only when there's an accident or something happens to a patient. And then they have to sequence that very quickly. They have to do all of that in one moment, and all of those people have to be just ready to go in that right spot, somewhere in the US most of the times. And Yes, they can do some very, very interesting and intriguing research, but when you cannot scale it up, when you cannot do, thousands of compounds, you can just do the one or two that are at the end just before the clinical trials. So yes, you can avoid the failure in phase one probably, but maybe you left something at the very beginning. And
Okay.
and it's impossible with those systems to do it. So the misconception, not the misconception, is really that people see this as those two extremes, and we believe that the middle is really much more. So we can do much more complicated models, but still high throughput, and still give you almost as much as of a value like those extremely expensive models. So that's really.
Yeah, yeah, interesting position in the market. But for example, what challenges have you faced in communicating your value proposition to investors?
Yeah. Thank you for this question. Yeah. So this is part of your first question. What is the transition? Uh, because really, investors really just looking, like in this kind of business, investors are looking for traction. So if you are just starting to sell, or let me answer this question slightly differently. Um, in biotech, there are two business models. I know people will tell you, oh no, there are many others. But if you look at what is being invested in, there are really two business models, with very, very few exceptions. One is that you just develop a drug. So there is, for investors, there's this extremely high promise of billions, literally billions at the end. The success rate is very low, but if you find the one, that's a huge payback. It's a huge payday. Um, and the other one is just based on revenues.
Yeah.
And so that means, I know several companies that did it. So you can start very low, which is good. So you can start, the first quarter can be 5,000 euros, the second six, the third, eight, the fourth 12. So you can start low as long as you keep that increase very steadily. After 2 or 3 or 4 years, you will find an investor. So that's something that those two models are very, very common, well understood, and investable. The challenge we are facing is that we are in between. So on our side, since the market is so heavily, it's I wouldn't say it's a blue ocean, but it's something like a blue ocean, because yes, there is the main competitor, but you can talk with anybody in the cell culture
Yeah.
industry. They will tell
Yeah.
you
Right.
that they are looking for a good replacement and they cannot find it. So there is a huge openness of our clients to actually do that. But there is also, that's part of the science. So they are also looking for publications, and to publish it takes really years. They are looking for this validation. So after many years, we finally have the data I told you about gemcitabine. It's a really remarkable set of data.
Yeah.
So we finally have it. In the last 5 years, we have, we have sold, I would say, quite a lot of to single clients, but without that huge exposure, like without this, big market penetration, it's really tough to find investors that want, like, investors are very much afraid if they don't see the patterns they are used to.
Yeah.
So the pattern they're used to are those two, or you just add more and more and more and more, and then obviously that explodes, or you just have nothing, and then uh,
Boom. Yeah.
then boom. And in our case, it was just up. And so we tested, for example, different business models. We wanted just to sell the product. That's what we're actually doing right now. And we hired a business developer, and she's doing very well. And but that's what we're doing right now. So there is this history where we actually had some very, very good services where we suddenly had a lot of, maybe not a lot of money, but really, there was surprisingly well done years, like financially. But then we actually were looking for some strategic partners, because our product can be implemented into many other solutions. So we thought, like, our business development was really more focused on getting those big collaborations, but that didn't work, because they really, like, the big companies really want to work with big companies.
Yeah.
And that was the lesson we had to learn. And going back to your question with investors, the challenge is really to find an investor who sees this up and down as really experimenting with business model, because that's in our case what it really is. And like now we see that the business model we're having is really working. So on one hand, we are talking with those big strategic partners, but it's this is something that will take many years,
Yeah,
and we
right.
know about it, and they know about it. So investors have to understand this as well. But at the same time, we do increase the sales steadily. So we have that path as well. But we also work on getting services where obviously suddenly we have much, much more income.
Yeah,
But the we are like, we do work as a small, very focused CRO, so contract research organization, very much focused on 3D cell culture models, basically. But that is really the work that we're doing just to get to that hockey stick growth. Because once we get even 2% of the 3D cell culture market, by the way, the market is like $2 billion,
yeah,
so and it's growing like 20% from year to year. So there is a lot of room in the market, and we see a huge potential in really doing that. So really, the biggest challenge with investors is to find an investor who understands. So biotech is different than IT. So if you're looking for returns in 3, 8, 12 months,
yeah,
it's not biotech. But if you're an investor who understands biotech and sees the value in the science, and also there is this huge, I think, very important tendency of kind of following the leader. So if there is an investor in Boston that invested in this huge company from Boston, it's much easier for other investors to join.
yeah,
And so this is really the more human side of doing the business.
right,
But
right.
yeah. So that I could talk about it for another few hours. So let me just
Yeah, right.
stop at this stage. Yeah.
Yeah, and how do you bridge the gap between the drug discovery and clear growth metrics?
Um, that's a very good question. And to be honest, we on one hand, I would like to tell you that we have we found a perfect method to do that. But to be honest, um, this is really something we are just working hard to communicate correctly to the investors, because we are not going to change our story. It is what it is.
Yeah,
um, and and that's why we are. Um, it's also interesting because when we are talking with investors, we're just very straightforward from the beginning. Like if you are looking for just for drug discovery or very clear growth, we do respect that. It's simply you're not going to find it with us. Like I would call ourselves a real startup, because startups, like, what is the definition? Like, what's the difference between another? Let's say you start a bookshop. Is it a startup or not? Like my definition, the definition of startup that I think is closest to reality is a company that really looks for the right business model for the product or services they offer. And to be honest, I would say exactly that's what we do. So we are like the business model of just selling. It works, but it will require a slower growth. The business model that we are now implementing on top of the sales of the product, which includes services, it speeds up significantly. And at the same time, we do work on those long-term goals, which are the strategic partners. And obviously, for investors, that should be also, kind of attractive, because probably those strategic partners could be future acquisition partners. So, in like, if you look at it from, I would say, first principles perspective, everything looks good.
yeah,
It's really about the mentality, about finding the right people, about being yeah, yeah, finding the right people, and being able to trust on both sides, and how to say that.
yeah. So being a real partner,
Yeah,
basically.
exactly. And but really understanding that this is a long term game.
Yeah, yeah,
But long
100%.
term. So to be honest, like yes, it's long term because we are already on the market for 9 years. But if you look at the margins we have, and if you look at the size of the market, which is basically still 95% of that is using solutions that are simply failing. There is this huge potential, but there has to be an investor who just understands it and is ready to go.
Right, 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 right.
Yeah.
And now, looking at a broader industry, what key trends are you seeing emerge?
So I mentioned that partially. So really, the trend that is really good, like on our side, is really one of the biggest perspectives that we see is what FDA and EMEA, so Food and Drug Administration in the US and the Your EMA, European Medical Agency, they really both of them push towards non-animal solutions, it's not just about saving animals, which obviously I do support entirely, but sorry, but it's I just had a post like I think 3 months ago. It was my best post on LinkedIn with like 70,000 views, something like this, uh, where I was citing data from 90s that very few people know about, and it was reposted like 20 times so far. So I'm not a social media guy, but I was really surprised. And this data is really striking because that data is really focused on toxicology studies,
Yeah,
but they are showing basically zero correlation of animal toxicology studies with human toxicology studies. And I'm not saying there is like I actually got quite a few very interesting comments from people from the toxicology industry, telling me that well, if you there is other way to interpret the data. So if you look at the raw data and there you standardize it in a slightly different way, there are ways to extract something. But they all basically agreed that there is a huge problem in translation in toxicology. And that's really one of the main reasons that we should think about when considering using animal models,
yeah,
because they can give you equally wrong impression of your asset to a 2D cell culture. I was on a one conference where one of the famous professors actually I think he was 100% correct. So he said something like, if you were a mouse and you had a cancer, we could treat you today with anything. Like we have so many drugs that can cure cancer in mice and that have no effect in humans. And that's why using advanced 3D models that use human cells, sometimes you have to use primary cells from the patients, but sometimes also cell lines have a good potential. So it really depends on the stage of what you're doing and the exact scientific question. That can, if done well, it can have a huge impact. So really, the trend, hopefully, and it is really going in a good direction. That's a huge push towards using advanced 3D models, and also from the regulatory perspective. So we are also talking with EMA, so European Medical Agency, to be able to allow our clients to use our assays. So whatever we deliver to our clients will be then accepted by EMEA. So
right.
that is really the trend that I see as really very, very hopeful. Uh, and and the good thing is also that it's actually progressing quite quickly. So like 2 years ago, obviously, okay, we all hear about that for many years, but 2 years ago I heard about the first regulation, and just a month ago I heard that they will not fund things that include only animals. So that's a really huge step. And with that, I'm very optimistic towards the future, but yeah, we have to get there.
Right, right. And as you wrap up, what's next for your research and exciting developments on the horizon?
So on our side, the example, like scientifically, the example I gave you with those drugs being actually almost on the same concentrations like in patients, especially in comparison to what 95% of the market does, we actually want to expand that very significantly. At this moment, we have it on 3 types of pancreatic cancer, and we expanded that to like 14 different drugs, and one of the 14 was actually the false negative. So we found a drug that actually works, where in 2D cell culture doesn't work. But then we found out that this drug, actually somebody did it, like a year or 2 years before us, because it's already in phase 2 clinical trials. So it's actually very good that we were able to pick it up, although
Yeah.
we are not going to make money of this discovery. Um, and that's really scientifically ahead of us, and we want to expand that. We want to be able to offer to our clients not only those few pancreatic models. We want to do that on all solid tumors, so it would be probably lung cancer, colon cancer, breast cancer, all of the big ones, to be honest. And then obviously we with our technology, we see that it translates very nicely to other solid tumors.
Right.
But from the business perspective, we're actively looking for clients, and we are fundraising. We have several discussions with investors. Just a few months ago, we closed a small round. So it's a very dynamic moment of company development, because many things are happening. And yeah, and it's something we will be looking back at in a few years from now.
Right. Right. And for listeners who want to learn more or potentially work with real research, what's the best way to get in touch?
I think the best way to get in touch would be on my LinkedIn. So you can find me on with my name, Marcin Krzykowski. And that would be really the easiest to reach out directly to me. If you want to learn more about what we do, there is our website is real-research.com. And you will find lots of scientific information there, although we are constantly updating because there are new publications coming up. We are just making protocols for our clients. We have a set with videos where we do
Yeah.
you can look at what we do, and you can always reach out to me or our business developer Elena, who we are constantly talking to people outside, and looking for a good way to really solve the challenge that you asked about, and to make that progress as quick as possible, because eventually there are people on the line. And I would just share maybe one experience that we had. We had a colleague from one of the companies that we collaborate with, and it was really something. It was really striking, because he was working on a very specific type of cancer, and he actually discovered a very nice combination of drugs for that type of cancer. And unfortunately, he got this type of cancer. But since he was a real specialist, he spoke with his doctor like an equal. And he told his doctor that for this type of cancer, he actually sequenced this and he knew exactly what is happening. And he told this doctor that there is a drug combination that actually works for exactly that one. And unfortunately, it took the doctors, I think, like 3 months or 6 months to do it, to get those drugs in house, because they were not standard drugs, and that's really the thing. And they worked, but that was just too late. And you just feel like it's a situation that it looks like the cancer was fighting back with him as a person
Yeah.
trying to fight the cancer. And unfortunately, he was really close to getting that if he got it sooner. But I think everybody has those stories around them.
Yeah.
So there is obviously, yes, we are focused on the business, but there is also this very human side of what we are trying to fight with.
Yeah, yeah, that's very interesting. And thank you for sharing those insights about you, the company, and the whole story. And thank you for
Thank you.
this amazing podcast session.
Thank you for reaching out, and all the best, Alexander. Have a good day.
Yeah, thank you. Have a great day. See you in the
You
next
too.
episode.
Bye bye.